Okay, welcome everyone. Thank you for taking time away from the World Cup. I think that's what's going on in the world today to make time for aquaculture. My name is Easton Kaboshik. I'm excited to welcome you here to the Soy Aquaculture Alliance quarterly webinar titled the Soy Research Trifecta, which now I'm realizing I hope I'm not considered one of the three in the trifecta. We certainly have the two experts here today. So I'd like to welcome Jacob Bledsoe. Hopefully I'm saying that right. Jake has been a great connection in the industry and a good research partner, currently serving as assistant professor and extension specialist in aquaculture at the University of Idaho. And then we have Marina, postdoctoral research scientist and working on one of our research projects through also the University of Idaho. So I'll have them introduce and speak to their backgrounds more. But on behalf of the U.S. Soybean Checkoff and the board of directors of the Soy Aquaculture Alliance, welcome and thank you so much for making time. We are here today in large measure to the 13 members of Soy Aquaculture Alliance. And really quickly, I want to list off the states in no particular order, but slightly alphabetical. Illinois, Iowa, Kansas, Kentucky, Michigan, Missouri, Nebraska, Ohio, South Dakota, Tennessee, Virginia, Minnesota, and then our newest member, Arkansas. So this webinar is made possible by those soybean producing states and the United Soybean Board. So on behalf of U.S. soybean farmers, thank you for making time and thank you for all the good work that you do in the aquaculture space. Couple housekeeping items. I'm sure many of you have attended a webinar before. If you have questions, please don't hold or wait till the end. There's a Q&A function in the Zoom webinar app. You can enter those at any time, and we'll make sure to answer them as quickly as we can. We're going to have our Q&A session at the end, but we'll try to answer those throughout. This is also being recorded, so for those of you joining us after, we had about 85, I believe, 85, 90 registrations for this webinar. If you're joining us after, again, thank you and welcome. This will be shared by email, and hopefully we can distribute this a little bit further on social media and beyond just our hour together today. So with that, I can't remember who's going first, if I'm being honest. I think it's Jake, but all right, brilliant. So Jake, let's toss it over to you, and please reintroduce yourself and jump right into the topic.
Yep. Thanks for the introduction there, Easton. Let me pull up my slides here real quick. Can you just confirm you're seeing things good on your end, Easton?
Yes, sir.
All right, great. So yeah, like I said, thanks to Easton and the Soy Aquaculture Alliance for this invitation. It's really an honor and a great opportunity for us to be able to kind of highlight some of the work that we've done. So I'm going to tell you today about our program. And as Easton said, I work for the University of Idaho. I'm an assistant professor there, research faculty, and then also the aquaculture extension specialist. And sort of this research trifecta that we kind of framed this talk as. The goal here is to actually kind of go through three little stories with you to kind of tell you how important soy's been to aquaculture and kind of the work that we've done at the University of Idaho to kind of advance that and get its implementation, more widespread. So the first talk I wanted to talk about you guys with today is kind of just laying the groundwork. So I want to give background on kind of a 25-year collaborative breeding program that we've worked on that's been based at our University of Idaho facility. It's down at the Hagerman Fish Culture Experiment Station. This has been a collaborative breeding program with the United States Department of Agriculture through their Ag Research Services. We've had continued funding on some of the mechanistic research throughout this whole breeding program from Soy Checkoff, from SAA. So again, this is just kind of a great way for us to bring this full circle, an opportunity to kind of show the overview of the work that we've done over the last 25 years. So we'll get started there. And then after that, as Easton said, we'll have two more talks of some recent research that SAA's thankfully funded for us. I'll pass things off to Marina after I'm done with the background on our breeding program. She'll talk about some of the great research she's doing on anti-nutritional factors and gut health. And then you'll hear from me again talking about kind of some of our early leading work on using machine learning to kind of help us in aquaculture as well. So without any further ado, I'll jump in here. So like I said, want to start off talking about our breeding program. So at our Hagerman Fish Culture Experiment Station, over about 25 years, starting in the early 2000s, we've been focused on trying to improve trout to have better performance on an all plant-based diet. And soy, of course, is the largest component of that. So, like I said, this work's taking place. I don't know, Easton, are you able to see my mouse if I reference things on the slide? Are you guys able to see if I kind of point or?
I don't think I'm seeing it right now.
Nope. Are you
Oh, yes, I am.
seeing?
There it
Yep,
is.
there you go. All right, there's a pointer function. So yeah, this is our beautiful Hagerman Fish Culture Experiment facility. So we're based down in southern Idaho. The great place about this location is that within about a 20-mile radius of our facility encompasses about 70% of the domestic food fish rainbow trout production in the country. So we're located right in the epicenter of southern Idaho's aquaculture operations and production. That means we're utilizing the same water that the industry utilizes. We're very close and able to collaborate and work with those producers and some of the other stakeholders. So that location is a big hallmark of what's allowed us to have the great results that we've had. So you can see our beautiful facility here. This is kind of our small little brood facility, and then we've got a nice large wet lab over in the corner here where we're able to do all of our phenotyping and selecting. So now we'll dive into kind of the rationale and motivation for this breeding program. So as I mentioned, this kind of really all started in the early 2000s, and the motivation was that we think about back to, even the 80s, 90s, and early 2000s, we had this groundswell of the blue revolution. So we were all told that aquaculture was the future. It's the most efficient way to raise animal protein. It's a very heart healthy protein to feed to consumers. So we need to increase aquaculture. However, traditionally at that stage, all of the aquaculture diets were very heavily reliant on these marine proteins and really even marine lipids. So diets were heavily based on fish meal and fish oil. Of course, there's some unsustainability both economically and environmentally there. So if we're looking to grow aquaculture, being traditionally reliant on those marine feedstuffs was very difficult from both a price point standpoint, the predictability of the supply, and it just wasn't a sustainable way to make the feeds in that growing industry size. So as we continued to increase soy inclusion levels and other plant proteins, that seemed like the very clear answer, right? And here in America, we have an excellent soy market set up. We have some reliable feed prices, and the supply is there. So it was an obvious choice for us to include into diets. However, those very early iterations were met with some production constraints. So those very high soy diets back in the 2000s that we started formulating, we'd unfortunately see reduced growth rates, poor feed efficiency. It turns out mechanistically that was all kind of based on its effects on intestinal health. Some of this like allergic, irritable bowel disease type phenotypes that we saw in those high soy diets early on. And then, of course, if you're having abnormalities in digestion, that can have concomitant effects on the water quality as well. So these high soy inclusion diets were a great solution for us to be able to grow our industry, but we were met with some performance challenges, particularly in these really carnivorous salmonids, things like rainbow trout or Atlantic salmon. We can think about things like tilapia and carp and those more omnivorous, herbivorous species. They're already well amenable to the soy diets, but it was these more carnivorous salmonids that we had a little bit more work to do yet ahead of us. So kind of the rationale of our breeding program was, we've started in the 2000s putting so much emphasis on novel feed ingredients and reformulating diets, trying to find the sweet magic ingredients that we could use to match the abilities of fish meal. But the rationale behind this program was let's not work just on the feed formulation side. Let's also work on improving the fish. So let's try to match this fish to feed compatibility and really get the harmony there. So we all know probably on the feed side, there's been a lot of innovation, various ingredient selections, from looking at regionality. Are there different soybean meals from different regions of the world, U.S. versus South American soy, looking at the differences between the sourcing. Of course, there's been, lots of focuses on ways to process and reformulate the ingredients. So do we just use soybean meal, soy protein concentrate? Do we take it all the way to an isolate? Are there other, processes that we can do to that? Things like fermented soybean products, where we can kind of change some of the nutrient profiles. And then, of course, just continuing to work on these formulations, adjusting macronutrient levels, micronutrient levels, and then, of course, even adding in some supplementations. So as you'll hear probably in Dr. Subramaniam's presentation, the optimal amino acids and fatty acids to supplement into these novel formulations, but also looking at, are there some micronutrients that we can add in to kind of help bump the performance on these? But been our job kind of to take on this other genetic component. So many people across the world, especially here in the US, have been working on improving the feed ingredients and the feed formulations. One thing that we're relatively unique on is our focus on improving the fish itself to be more amenable to that diet. So again, kind of trying to take that harmonious approach of continue to work on the ingredients and the formulations, but let's try to make the fish match that as well. So what we've done is really just tackle on a very classical Mendelian kind of breeding program. So the idea was to just evaluate families for their performance on these novel, soy-based target diets, and then just do kind of classical performance. So look for the strongest performers, the fish that grow the best, have the least amount of intestinal abnormalities. Repeat this generation over generation. And again, just like any agricultural selective breeding program, hopefully make good performance gains. And again, ultimately get down to this fish to feed kind of compatibility. And the idea being that, this has been a really successful tool and harmonious for our program. But I think thinking about feed formulation and selective breeding and kind of the complementarity of these tools really has applications outside of soy in aquaculture as well. So just keeping in mind the harmony of those two different approaches. So now that we've kind of covered the rationale, to kind of quick give you the history, background, and current aspects of our breeding program. So 25 is a nice round number, so I always say that we're in our 25th year, but technically it's actually our 24th year of selection that we just started this year. So our breeding program was started back in the early 2000s in 2002. Again, like I said, this has been a collaborative breeding program from day one. So the USDA, the Agricultural Research Services, their stakeholders let them know that this is an important area that we need to work on. We need to figure out ways to improve terrestrial grain inclusion in these sustainable aquaculture diets. So this was motivated through the Trout Grains Project, a USDA CRIS funded project. They brought in geneticist, Dr. Ken Overturf was hired, and he led the program from its beginning in the early 2000s, leading all the way until his retirement last year from the program. So the initiation of this breeding program, we actually took 9 different improved lines of rainbow trout. Some of these were hatchery stocked, some of them were slightly improved commercial lines. But the idea there was to phenotype all of these various relatively improved and controlled breeding lines of rainbow trout to try to find that phenotypic advantage and that tolerance of a high soybean-based diet. So in the early 2000s, they integrated 9 different selected strains and then basically just started working on different feed formulations to challenge those strains and look for the winners, right? Again, very classical agricultural animal selection. So in those early years, the approach was to make a fish meal free plant protein based diet. So completely eliminating fish meal in those early years was pretty difficult. We were looking at early formulations, probably about the 15% soybean meal inclusion level, but then adding on soy protein concentrate and some of the other corn proteins to balance out that protein replacement. But in those early years, we saw a lot of challenge. Like I mentioned earlier on, there was reduced growth, reduced feed efficiency, kind of this phenotype in the gut, similar to humans that have ulcerative colitis or irritable bowel syndrome, with nonspecific inflammation and enteritis. As they continued to advance that, though, into the later years, they were able to actually make some improvements to that germplasm. So, in about the middle of our program, they were able to start actually increasing the soy inclusion levels. So, those early 5 to 10 years, a couple rounds of selection on that germplasm was able to get fish to where they were able to tolerate that new formulation. So, in those later years, we were able to start pushing that up to a 20-25% soybean meal inclusion. And then finally, here into the expanded research phase that we most recently are kind of getting out of. Once we've created this model, we've now got a fish that thrives on this high soybean-based diet. We've now been able to do a lot of mechanistic studies to try to understand the biological mechanisms that have kind of conferred to this germplasm to allow it to perform so well on that soy-based diet. And I want to say this has been something that's been a lot of work funded by the Soy Aquaculture Alliance. Some of these mechanistic studies looking at gut health, metabolism, its effects, unintended effects on immunity, and then of course looking at how our selection has changed the nutrient utilization in these novel feed formulations. That brings us to today. So like I said, Dr. Overturf retired from the program last year. I'm currently leading our breeding program, and at the moment, we've been focused on kind of the same selection trait. So the only new item that I've brought in is we're trying to change kind of the environmental selection components here. So we're now doing selection in a common garden setup where we're able to control for environment using genetics a little bit better. So again, to kind of give the overview, this is a very classical breeding program. We take our various families, originally brought in from 9 integrated lines, now kind of considered our nucleus Hagerman breeding program. We select those fish on the selection challenge diet. Again, started out early years at about a 15% soybean meal inclusion. We're now up to a 25, and now the most recent this year's formulation is at 30% soybean meal. So we've really upped the inclusion levels of soy on those diets. Feeding those challenge diets, measuring the performance of the individuals, putting in kind of some weighted metrics on the family values and trying to manage inbreeding, but basically just looking for the best performing fish each year based on that challenging diet. From that, we select our brood and then continue year over year. The one kind of abnormality here is just that rainbow trout are a 2-year maturing fish. So we kind of, over the 25 years, really had 2 year classes. We had our odd and evens. But then, of course, to be able to get that hybrid vigor, thinking that we have two improved soy selected lines with odd and even year, we've had two specific times where we've actually done those crosses where we've take 3-year-old males and put those onto a 2-year-old female. So then we've even gotten additional hybrid vigor by combining our odd and even year selection classes. So now that you know a little bit of the background, I'll tell you a bit more about our challenge diet that we use for our selection. Like I said, this is a completely fish meal free formulation, 100% based on plant proteins. So altogether, between soybean meal and soy protein concentrate inclusion, we're up to nearly 50% soy-based diet. So you can see that, from those early diets in the 2000s, we've now got a diet that's nearly half of soy between the different feedstuffs that we use, and the fish are thriving on that. It's a pretty typical formulation for trout. We're targeting about 40% crude protein and about 20% lipid. I will say that this particular program, we do still include fish oil in this formulation, although we have had some spin-offs where we've actually started selecting fish for complete replacement of plant proteins and plant oils as well. So, yeah, we have some spin-offs there, but this diet in particular still has a good bit of fish oil in it to bring in those omega-3s. But again, this has been the whole kind of key to our selection program, just hitting them with this continuing selection challenge diet. So now that you know how the breeding program was established and kind of how it works, I'll talk to you about some of the outcomes that we've gotten. In terms of our production response, thinking about, a farmer's perspective from aquaculture, the biggest thing here is that we now got a fish that we could get to market in about 1 month's less time than what any of the other commercial lines would be able to achieve. But this is also while on that cheaper, more sustainable plant-based diet. So we could compare our strain, and we do this often, nearly every year. We take commercial, control lines that are, commercial germplasm used in the U.S. industry, raise those up alongside our strain, both on a classical fish meal based diet and a more modern plant-based diet. And again, by the end, we're able to see within a month of time that we can save getting our fish to a market size. But that's even comparing them to a commercial line that's on this more gold standard fish meal based diet. So able to save the producer's time getting to market quicker, but also on a more economically advantageous diet. So had a lot of gains in terms of the overall phenotype. Mechanistically, again, some of this has been through the help of soy aquaculture. And Marina, my apologies, I realize I'm cutting into your time already here a little. But we have dove into some of those mechanisms in the later years. And so what we found is part of the reason these fish are able to grow so much better is they just don't have that intestinal abnormalities that we saw in early unselected lines when feeding these high soy-based diets. So on the left side here, we have just regular H&E histology stains. We have PCNA, which is a proliferation marker, and then we have fatty acid binding proteins. Same slides are over here on the right side for our Hagerman selected slide. And I don't have enough time to walk you through all the histology, but basically we have some abnormalities. We have reduced proliferation, immune cell kind of turnover, and clearly reduced fatty acid binding capacities in the gut here. Whenever we look at our strain fed that same diet, we see a very classical, good intestinal phenotype. So they're maintaining their intestinal architecture. There's a lack of this intestinal inflammation response that we see, and we don't develop this soybean meal induced enteritis that kind of became the hallmark of soy diets in the early 2000s. In addition to that intestinal mechanism, we've also found that we had some unintended improvements in other areas too. So, this is from some recent work doing proteomics. Again, if we look at our more susceptible commercial line, doing our classical design with fish meal and plant meal based diets, whenever we feed that commercial line this high soy based diet, we see signs of intestinal damage, like shown on the previous histology slide. We've showed at the RNA level and as well as the protein level, so things like transcriptomics and proteomics, we found innate immune responses get triggered and high inflammatory response. You see DNA damage and cellular stress responses, and really you eventually get this soybean meal-induced enteritis that causes a whole body systemic inflammatory response. But again, whenever we do this in our selected line, we're not seeing those deviations from kind of the norm whenever we feed this improved soy-based diet. Recently, from I would say both from transcriptomics and from proteomics, we have been able to find some markers. The transcriptomic work has found that fatty acid binding protein is consistently different between these two strains, so it shows that our selection has been allowing these fish to more adequately absorb those nutrients. And then this NLRP1 is an immune marker from some of our recent proteomics work that's kind of a master regulator of immune response. And we showed a reduction of that in our selected line, even fed that plant-based diet. So it's basically able to master regulate the response and have a more normal phenotype. Our little bottom plots here just show that we also have improved performance in disease challenge. So our strain has only been selected for plant-based tolerance. However, whenever we challenge it with IHN, so one of the most prominent viruses that hits trout, it does middle ground, and this is in comparison to a strain that was selected specifically for IHN resistance. But it does better than the commercial line that had no IHN resistance selection. Similarly, if we compare this to a strain that was selected specifically for resistance to a bacterial pathogen, the causative agent of bacterial cold water disease, again, whenever we compared it to these commercial lines, we saw that our selection strain still had better survival there. So not only have we produced or improved our performance on soy diets, but we also have got this improved immunological outcome as well. So in terms of what our next steps would be, next phases, the idea now is to kind of preserve some of these phenotypic gains and the advantages that we've made on the soy selection side, but start to expand that genetics and improve other traits as well. So we want to work very carefully to not lose the selection advantages that we've built, but we want to bring in some new genetics as we're starting to bottleneck over these 25-year program, and then also bring in some new traits, and then working with new technologies, things like digital and AI phenotyping, working very closely with our industry stakeholders to identify new targeted traits for improvement. The idea is to take these selected gains, integrate them into a new germplasm, and then continue to select on for further improved traits. Again, looking for the overall outcome of improving that fish feed compatibility and the performance that we see from the fish on that advanced diet. All right. So with that, again, I meant to kind of give that as just kind of a broad overview of our program. I'd be remiss if I didn't acknowledge the many people on both the USDA side and the University of Idaho side over the last 20 years that have made this program really the outstanding breeding program that it has been. So now that was kind of meant to be the background. This has been an emphasis and really like a thematic theme for our program. So the next two talks will dive into some of our more recent research projects funded by SAA and talk to you about the advances that we've made from those. With that, I'll pass things off to Dr. Marina Subramaniam.
Thank you, Dr. Bledsoe. I'm going to go ahead and share my screen.
And take your time, Marina. Sorry, I used up a few of your minutes. I'll pay for it on my next presentation. So.
You're totally fine. Okay, I believe everyone can see that. Okay.
Good to go.
Perfect. Thank you. I'm Marina Subramaniam. Before I start my introduction, I just want to apologize. I'm getting over a really, really bad cold. I've been coughing a lot, and if I get into a coughing fit, I will mute my mic so I don't blow anyone's eardrums out. So I'll make sure to do that. Okay, so I'm Marina Subramaniam. I'm a research scientist here at the University of Idaho at the Hagerman Fish Culture Experiment Station. My supervisor is Dr. Small. My research is mainly focused on nutrition and nutritional physiology, understanding how alternative protein ingredients or alternative feed ingredients affect intestinal function. And I use a specialized technique to study that. Also, I am Canadian, so today's July 1st. It's Canada Day, so I just wanted to wish Happy Canada Day for me to all of you guys. Okay, let's get into my presentation, which is physiology-based research to understand SPMIE or soybean meal-induced enteritis and maximize soy nutrient value for fish. So briefly, my outline for today's talk starts with current challenges in fish nutrition. Again, a very broad statement. I'm going to focus on specific knowledge gaps that we hope to answer with our research grant, which is from the Soy Aquaculture Alliance grant. And I'll talk a little bit about that. Then I'll get into a background of digestive physiology to kind of better understand what I'm studying and how it looks like and the specialized technique that we use. Then I'll get into the objectives of our grant, present some results we have so far, and what implications our research program has for soybean and aquaculture industries. Okay, so current challenges in fish nutrition. And again, a broad statement, I'm going to focus on feed, right? Feed accounts for most of the production costs. And the limiting factor here is protein coming from fish meal, which is expensive and unsustainable. Therefore, we've branched out to alternative protein ingredients coming from plants, such as soy, insects or insect meal, as well as single cell organisms such as yeast. Now, when we feed these alternative protein ingredients, such as soy, we measure metrics such as growth performance and health. However, how does this work on an underlying mechanism? How does it affect intestinal function? If we can better understand that question, we can answer questions such as, how do these feed ingredients work? Why do they work? Why don't they work? And these are some of the knowledge gaps we hope to answer with our program or with our research. Specifically, when you look at soy, how does soy affect nutrient utilization? I have an animation here of a nutrient getting absorbed across the intestine. How does that mechanism work and how does soy affect that? How does it affect fish health and welfare, right? Does it enhance disease resistance? How does that mechanism work? The effects of anti-nutritional factors. ANFs are components in soy that provide a protective mechanism for the plant, but can cause adverse effects when fed to salmonids. But how does that mechanism work? And how does that affect nutrient absorption? So all of this has effect on gut function or intestinal function, which ultimately affects growth and health of production fish. If we can better understand these knowledge gaps, it can have positive implications for the industry. We can apply this to the soybean industry, selecting for better soy, especially mitigating ANFs that cause these adverse effects. It has implications for fish feed and aquaculture production, helping us reevaluate nutrient requirements for fish, and of course, increasing or maximizing soy inclusion, especially for our carnivorous salmonids. So getting onto our grant, we're looking at soy, alternative protein source from soy-based feed ingredients such as soybean meal. However, high inclusion levels for salmonids has resulted in hindered growth and affected digestive function. Specifically, it has been shown to impede nutrient absorption, but the underlying mechanisms of that is still unclear. So when we talk about nutrient absorption, at least for our grant, we're going to focus on protein. Amino acids are a building block for protein, and we are focusing on methionine, a type of amino acid. Methionine is considered a limiting amino acid or an essential amino acid. Excuse me, I think I'm back. Methionine is an essential or limiting amino acid, means it has to be acquired from the diet. And soy diets tend to be low in methionine. So we want to study methionine absorption because it's crucial to investigate its absorption in commercial fish species and potentially its impedance by ANFs and how does that work. So with our grant, we hope to be the first. Oh no. We hope to present the first definitive molecular characterization on the impacts of SBMIE and targeted ANFs on nutrient absorption and gut barrier function in Atlantic salmon and rainbow trout. Before I get more into our grant, I want to talk about digestive physiology and what I'll be specifically measuring. So, understanding the underlying mechanisms on how soy-based ingredients affect growth, performance, and health, we need to start with the gut, right, which ultimately affects growth. The gut is where the nutrients are digested, they're absorbed, and that's how these fish use them. So, we have to start with the digestive tract. You can call this the gut, you can call this the gastrointestinal tract, the GIT, it's all the same thing. And we can characterize nutrient digestive and absorptive mechanisms in response to soy-based ingredients. And the key point here is that if we understand how these mechanisms work, we can take advantage of these mechanisms to offer solutions and suggestions to different industry areas, such as feed formulations, feed processing, and mitigation strategies that can result in increased inclusion levels of soy in fish feed. That's a very great statement, but how do we study that? Well, for our research group, we use something called the Ising chamber. And before I get into the Ising chamber, I'm going to explain how nutrient absorption works on a molecular level for those who are unfamiliar with it. So we start with our fish. This is rainbow trout. I've dissected the intestine out of that trout, and I've divided the intestine into 4 intestinal segments. So we have the anterior proximal and the posterior proximal, which is right after the stomach portion. We have the mid-intestine, and we have the distal intestine, which is the end of the intestine. And for the purposes of our grant, we're focusing on the mid-intestine. I might have to. Okay, I'm back. All right. So from a molecular perspective, these are the cells that line the intestine, right? We have the apical or lumen portion of the side of the cells. This is where all the digested feed, the nutrients, all those products are present over here. The basolateral side represents the blood side. Nutrients are generally absorbed from the apical to the basolateral side. So we have amino acids, which are the building block for protein. We also have monosaccharides, which are the building block for carbohydrates. We also have a bunch of ions, negative and positive ions, that are also present on either side of the intestinal lining. So how are nutrients generally absorbed, right? They can be co-transported with an ion through these transporters. These transporters are what takes these nutrients or an amino acid from the apical to the basolateral side. And then it goes to the blood, and then it goes off to the liver where it's further metabolized. Methionine transport works similarly. So we call this sodium-dependent methionine transport, where methionine is getting transported across the intestinal lining along with the sodium ion. When we take this into the Ising chamber, how do we measure that? We measure it as short circuit current. The movement of ions create current. We can indirectly measure methionine movement going across by directly measuring sodium movement going across. Additionally, with our grant to measuring sodium-dependent methionine transport, we're also looking at gut barrier function. And this is specifically looking at the space between the cells. Now, that space between the cells, or the function of that, is highly regulated. So that space can be really small in certain intestinal segments, or it can be really big. And that space can be manipulated based on what ingredients and et cetera that you feed. And we can measure this on the Ising chamber as transepithelial electrical resistance, or TEER. So if the space is really big, the resistance will be low. If the space is really small, the resistance will be high. Okay, so getting into our Ising chambers, we start with our intestine. Again, I've divided it into 4 intestinal segments. Again, I'm focusing on the mid-intestine. So I cut that segment open and I expose it as a flat sheet on this insert. And the area of exposure here is 0.3 centimeters squared that we find work best for fish. And from there, I place it into my Ising chamber. So the Ising chamber, I'm being very biased. It's a very, very cool technique. It basically, we can take the gut sections out of fish and keep them alive outside of the fish and study isolated mechanisms within that segment. So it gives us a lot of information. So when I place the insert into the chamber, the apical side faces the left and the basal side faces the right. This is bathed in a teleose buffer or teleosaline that mimics the internal environment of the gut. We also have a gas input that mixes the contents within the well, also keeps the pH of the buffer constant, and this is all placed into a jacketed chamber or stand that controls the temperature of the chamber. So here, this would be the housing temperature of our fish, which is around 15 to 15.5 degrees Celsius. When I measure methionine transport, I'm basically adding methionine to the left side and measuring its transport across to the basal side, mimicking what would happen inside of the fish. So for the objectives of our grant, we have two major objectives. The first line is to characterize baseline gut barrier function and methionine transport. And why we want to characterize baseline is because very little is known on how methionine transport and gut barrier function work, especially in Atlantic salmon and rainbow trout. A lot more work has been done in rainbow trout, but not as much in Atlantic salmon. So I need to characterize baseline transport before I can see how those transport and functions change. And we also looked at isolated anti-nutritional factors such as lectin, saponin, and beta-conglycinin, and we used Ising chambers supported by qPCR or gene expression. Therefore, the second objective builds on the first one, where we would conduct basically a 12-week feeding trial where we used a soy-based and a fish meal-based diet in both Atlantic salmon and rainbow trout to assess changes to methionine transport and gut barrier function. And overall, what we're measuring here is short circuit current and intestinal integrity. And these are the two main results I'll be presenting today. So for short circuit current, we'd be measuring sodium-dependent methionine transport. Again, the movement of ions create a current. We can indirectly measure methionine transport going across by directly measuring sodium moving across. You can also look at total methionine transport. This is sodium-dependent and sodium-independent, meaning methionine can also be transported across the intestine without sodium. And there are transporters present for that. But I won't be presenting those results for today. And of course, gut barrier function, looking at that space between the cells measured as transepithelial electrical resistance on the Ising chamber. So again, I'll be focusing on the first and the third point. Okay, so let's get into my project. I work with rainbow trout and Atlantic salmon, and we're focusing on the mid-intestine section. Oh my goodness. Okay. So I'm
You're
focusing
great.
on the, thank you. So focusing on the mid-intestine segments, let's get into the interesting results. So very briefly, transporters work where they transport a nutrient going across the intestine. You can characterize these transporters, meaning you can see how fast they transport them, how slow they transport them. Do they have a high binding affinity? Like, can they recognize very low concentrations of this nutrient? Or do they require higher concentrations so they have a very low affinity, right, for the nutrient? So there's different ways to characterize it, and that's basically what I've done. So very briefly in this graph, I ran a methionine gradient where I added methionine to that left side and measured its transport to the right side. And I ran a small gradient, so from 0 to 150 micromolar. That's what's listed on the x-axis. And as I add more and more methionine to the chamber, the current goes up, right? So I can measure that as change in short circuit current on the y-axis. And what's exciting for me here is that we got an S-shaped curve, which exhibits sigmoidal hill kinetics. Now, what does that mean other than the cool name? Well, sigmoidal hill kinetics suggests more than one transporter is transporting methionine at the lower concentration. Subsequently, I ran a higher concentration gradient of methionine, and here this exhibited Michaelis-Menten kinetics. This suggests that there's only one transporter population transporting methionine at the higher concentration. Why is that important? Hasn't been characterized before, right? So this is new. And what's interesting is that Atlantic salmon exhibited similar kinetics. So again, we focused on the mid-intestine at the lower concentration gradient. This also exhibited sigmoidal hill kinetics, suggesting more than one transporter population transporting methionine. Whereas at the higher concentration gradient, it exhibited Michaelis-Menten kinetics, again, suggesting one transporter population. Now, how do these mechanistic results apply to the industry, right? How would this apply for feed or aquaculture or soybean industries? Well, soy generally tends to be lower in methionine. So we supplement methionine into the diets. If we understand how saturated these transporters get, it can help us reevaluate and determine how much methionine to supplement. Are we adding more? Should we add less? Right? Additionally, and this is taking it one step further, when you do a comparison between species and you see similarities in kinetics on how they transport these nutrients, it can also help us reevaluate and determine nutrient requirements for different species. Maybe they're more similar than we thought, right? So that's the type of applications that we hope to get with our research. I also looked at a specific anti-nutritional factor. I'm going to show saponin. So saponin after methionine reached saturation, I added a dose-responsive saponin right after, and you can see here that we got inhibition of short-circuit current. What does that mean? As we added more and more saponin to the chambers, it inhibited methionine absorption or methionine transport. This is a raw trace from my experiment. So here is my methionine gradient, right? I'm adding methionine in, it's saturating out. Then I add saponin in, right here, and you can see the current drops. Interestingly, for gut barrier function, saponin did not result in any significant inhibition, which again, one reason for that may be a longer exposure time of saponin with the tissue, right? But we didn't see any significant inhibition. Interestingly, Atlantic salmon also showed the same results. So when we add saponin in, we did a dose response, and that also resulted in inhibition of short circuit current, again suggesting that methionine transport was inhibited. But with gut barrier function, we did not see any significant inhibition. So very similar results between the two. Methionine transport was inhibited, but gut barrier function, or that space between the cells, right, that intestinal integrity, did not seem to be affected, at least with the saponin protocol that we ran. So what are some implications for the soybean and aquaculture industries? So again, for challenges, right, we want to limit feed costs, especially when we could use sustainable, cost-effective alternatives, alternative protein ingredients such as soy. And by understanding how soy affects intestinal function, this can help us increase inclusion of soy in fish feed, as well as mitigate impacts of the anti-nutritional factors. And these are the knowledge gaps that we hope to better understand, right? How do ANFs affect nutrient utilization? How does that affect overall fish health and welfare? What are the mechanisms there? And we can isolate and eliminate the specific ANF components, which brings me to my deliverable, especially for the soybean industry and the aquaculture industry. If we can identify and characterize ANF effects on nutrient absorption, we can isolate and eliminate these harmful ANFs to help us maximize or increase soy inclusion in fish feed for salmonids. That's the end of my presentation. For acknowledgements, of course, I want to thank my supervisor, Dr. Brian Small, who's also the PI of this grant, as well as Dr. Madison Powell, who's the co-PI of this grant and has been a tremendous help throughout this project, as well as all of our wet lab technicians and our nutrition lab manager as well. And of course, the funding agent, Soy Aquaculture Alliance, for funding this entire project and allowing me to study something new called the Ising Chamber, and of course, the University of Idaho. On to you, Dr. Bledsoe.
The only bad thing about webinars is we don't all get to like clap for how well that was put together. So you did a fantastic job, and I really do appreciate the animations and breaking that down for a humble schmuck like me.
Thank you so much. I'm glad I was able to do it without dying.
Yeah, only 3 coughing attacks. You survived. Perfect.
Can you just confirm, Easton, you seeing my slides okay?
Yes, sir.
Very good. And thank you, Marina. That was an excellent presentation. We're very fortunate to have Marina in the aquaculture industry. Honestly, we brought her from some of the human medical space. She was working at a very well-known hospital before she joined our team. And you can see from the work she did there that she's been a big value to get all of her technical insights into aquaculture. I think we're going to see a lot of advances come out of her work moving forward.
Thank you so much. I appreciate that. Thank you.
Yep. Just kind of continuing on, talking about some of our recent research outcomes that have been collaborative with Soy Aquaculture Alliance. This is a project I just wrapped up a month or two ago, actually. So the idea here was to try to develop some machine learning algorithms to see if we could take a look at rainbow trout intestinal histopathology. So kind of the motivation for this, it really comes down to the fact that the histology has kind of been the gold standard for a way for us to assess how these alternative novel feed formulations are being, taken by the fish that we're feeding them to. So Marina just presented a really awesome new platform, this Ising chamber. I really do think this has a lot of potential to be one of the new gold standards for how we assess these new novel diet formulations. But up until the point of her kind of developing this and getting this information out, like I said, distal intestinal histology has really been the gold standard way for us to say, does this novel diet perform well? Does it cause any, intestinal abnormalities? The problem is that doing intestinal histology is really reliant on histology labs and then trained histopathologists. So you've got to cut your slides at the study level, but then send it off to a well-trained lab to be able to prepare your histology slides. And then the biggest limit has been finding a trained histopathologist, particularly one with some relevance to aquaculture and aquatic species. It requires specialized expertise. It takes a significant amount of time. These histologists are spending, hours or multiple hours per slides working on studies that require hundreds of slides. So it's just a substantial time commitment. And we need to have consistent traits. So each study, we often end up, whatever histologist is available, that's who you go with. And it kind of leads to some inconsistencies there. So that again motivated this idea of could we use some of these advances that we've seen lately in machine learning and AI to try to bite off kind of some digital pathology here and use some of those tools to make things a little more consistent and hopefully reduce some of that time and cost burden that's in this gold standard way that we assess novel diets. So to do this, what I proposed with the Soy Aquaculture Alliance, initially the goal was to kind of crowdsource slides from all over the world. We have a big network, as I talked about with our breeding program. We've been focused on this soy work for the last 25 years, so we have a global network of researchers working on the same task. So my goal was to crowdsource lots of slides from all over the world, but it turns out us researchers, it's pretty niche a lot of times to be able to share your slides. There's grant complications, anonymity of data. So we actually had a difficulty in getting the total number. So one of my limitations for this overall kind of outcome of this would say, just the total number of slides I've gotten. So we started with a 200 slide deck. After whittling things down, we had 182 slides that I actually used to train this model. So to do this, we had to first source these slides. These all came from internal University of Idaho slides, but I was able to make sure they varied across different life stages of rainbow trout. One of them is a very on-farm applied trial. So actually fish sampled from a raceway on a soy versus traditional diet study. And then the other two studies are more classical research like we do. So as I get to kind of some of the endpoint deliverables, I would just say here at the beginning, our biggest weakness probably is just getting a bigger amount of data to train this on. And the old adage in academia is garbage in, garbage out. By no means is this garbage, but more will only make the outcome a whole lot better. So, but it was really a great proof of principle and a learning kind of undertaking for us to learn a lot more about enteritis and using machine learning is kind of this new analytical method to figure out a lot about this concept. So to first off, we use kind of classical rubrics for describing intestinal enteritis. So things like the URAN, let's see, there was three different classical rubrics that we use from 2008, 2010, and 2012. Those different rubrics for how you described enteritis, they all focused on kind of some of these same phenotypes. So inflammatory cell infiltration, granular cells, goblet cells, looking at the architecture of the intestine itself, and then looking more through the epithelium for vacuolization or villi length and width. But there was disagreements in how those rubrics work. So we did a lot of work here to kind of summarize a new, what I want to recommend as a gold standard intestinal scoring rubric for our industry to use to be able to describe enteritis. And then we use the scores from that data after getting ground truth, trusted histologist, worked with a world-class histologist, Dr. Salvatore Frasca, and with his private company now, Aquatic and Investigative Pathology, to ground truth all these slides. And then that's what we use to actually train this model. So first off, a little bit of data kind of digging background. Across those 200 slides that we scored, of course, not all of them had enteritis, but whenever we split across our study from enteritis positive and enteritis negative, a first deep dive we wanted to do was, are there specific traits that we could even use to train this model on that describe enteritis just as well as the overall final definition of enteritis or not? So the first thing was just to take all of the phenotypes that we characterized in the histology slides and rank their effect size. So here you can see our mononuclear cell infiltration was really a real front runner here. And you can see all of our enteritis positive slides had a much higher severity of mononuclear infiltration, while all of our negative enteritis slides, other than a few outliers, had almost no infiltration. So we were able to find a strong biological signal of that mononuclear infiltration. There were also some signals here at the villi architecture level. So we do see some of that clubbing and shortening of villi here on the enteritis positive fish having kind of a wider villi. But at the length level, it really didn't wash out as well. So that showed us that in terms of our, other traits other than enteritis, that mononuclear infiltration was our strongest biological signal to also look at this in addition to just the overall enteritis yes or no. So in terms of how we actually undertook this model building, so we started off with these whole slide images. So this would be the entire slide. So literally a small glass slide. Some pathologists or histologists are going to put multiple images per slide, multiple sections. So this is a section that had three sections per slide. We built a Python computer science kind of algorithm that's able to go in and actually automatedly identify those sections, so you don't have to even tell it anything. You do have flexibility to say, is this a single section per slide, or are there multiple sections? Do I split sections out into different sample names based on metadata? So there's a pretty sophisticated QA/QC to be able to region of interest extraction and pull these slides out. After that, we do quality control on the whole image, and then we start tiling these. So tiling is basically breaking this whole image down into many, many subsamples. It's based off of pixel sizes and resolution. So these are kind of examples of some of the pixels that we would get from a whole non-amplified image, and then we break that down, go into the full 40x amplification view, and then tile those, and you can see kind of the subsamplings that we get. This shows high quality slides. Again, we have an automated algorithm that pulls out these medium or suspicious quality tiles, and then that can be manually curated. And then it automatically observes these bad tiles. So these are tiles that just grabbed some white area off this slide, and we exclude those from all of our training or data assessments. And then this just kind of shows you the ready to go analysis tiles that have metadata tied back to, was the whole slide enteritis positive or negative? So you really could call this a weakly supervised model, because we took some pre-trained models out there that worked on computer vision and then just used my tiled data and the ground truth histology scores to be able to train that model to be able to identify these traits on its own. So in terms of the results, the first task here was to look at binary enteritis positive or negative. We had some relative good success on this enteritis classification. You can see our breakdown over here from predicted, the kind of how things should have went. We should have complete agreement down this diagonal if the model performed exactly like the histopathologist. Unfortunately, we do currently, because I wrote this model to be very conservative. The idea here is that we don't want to completely replace a histopathologist. We want to do a first round quickly through this AI and flag slides that we think may be showing signs of enteritis. And then those, between these 22, between these 15 and 9 slides that the model called out, the histologist can then focus his time just on those 16 slides or 26 slides and reduce the burden, and then decide, were these actually positive or not. So again, more iteration can be done here to work on kind of some of the different computer science steps through this to improve this model. But at the moment, we've built an enteritis model that can, with 100% sensitivity, call out enteritis. The downfall is that its specificity is reduced with some of those false positives. But again, I wrote the model to make sure to call out that enteritis was going to be present. And we built really hard, kind of coding in there to make sure that we didn't have false negatives, where we did have a true enteritis phenotype, but the model falsely characterized it as healthy. So again, relatively early kind of outcomes, but very promising, showing the potential here for very automated enteritis description. However, enteritis alone, just saying enteritis yes or no, isn't always all that informative, and things have begun to get more nuanced as we see less overall enteritis. So again, I told you that mononuclear infiltration was our leading biological insight. So I then tried to be able to train a model to be able to actually score 1 through 5 on the mononuclear infiltration levels. So instead of just this binary trait, we're now doing an ordinal across 5 different categories. Turns out that's much harder. We can see that from the results here, the confusion matrix. The biggest problem here, to be honest, gets down to what I just mentioned, is that we're actually not seeing as severe cases of enteritis anymore. And that gets back to a lot of what I talked about in both my talk, my first talk, and Dr. Subramaniam's talk about the improvements that we've made to diet formulations, the improvements that we've made to fish genetics. Those classic severe cases of enteritis just aren't really being seen anymore. And you can see here from our predicted or our observed even, we didn't have severe 3 through 5 cases of severe infiltration. So that again limited our model in terms of having just the training data there. Again, the accuracy here, some would argue this is just about like flipping a coin, right? 50% chance of being right on your decision, but that's really not true because we're classifying across these ordinal categories. And again, we're making decisions from breaking this weakly supervised whole slide down into individual tiles. So whenever we actually look at this, it's really about 3/4 of the times of the decisions that the model is making, it's getting it right on this mononuclear infiltration. Certainly still a lot of work to be done, but it's very clear that this inflammatory signal was detectable just from this computer vision model. Without any sort of input, the model was able to learn signatures of inflammation and enteritis that we found here. These outcomes certainly need to be improved, and this model can't be thought of as a handoff replacement for histologists, but there's a lot of really valuable insights here on model training, and I think the future has a lot to hold for AI and digital phenotyping in other traits as well. So to kind of summarize, just inflammation was easier to learn at the large scale than the intestinal architecture. That has to do with my tiling approach. I think more advanced ways for us to take this model is actually trying to segment these models to where the model's smart enough to say, this is a VLI, this is the lamina propria, this is where a vacuole is versus a goblet cell. The amount of work and data that's required to train such a model is exponentially larger than what we undertook with this project. But I do think that my results do show that we will need those informed segmented models because, like I said, no longer in the 2000s we could have just said enteritis yes or no, and we had severe cases of enteritis. Today, the soy ingredient, the diet formulations, and the trout genetics have gotten so much better that we're now looking for much more nuanced biological kind of perturbations in there, and we can't just describe enteritis yes or no because most of the time the answer is no. But we want to know, has vacuolization reduced? Has goblet cells increased? We need those more granular biological kind of insights. So again, I think we're going to have to spend a lot more development time on the future iterations of these models to be smart enough to segment out that histology slides. In terms of what's really still required, I've basically said I view this as a proof of principle and kind of a first step. I think this is the first step in kind of a long journey, but it does show a lot of potential for this digital phenotyping. In the future, we just need more slides altogether, but I'd like to have those slides reviewed by multiple pathologists to make sure we aren't biased on one potential histologist. And again, I need to get more of these mild and severe cases. So if there's any aquaculture practitioners out there that know of very severe enteritis that they're seeing, give me a call because we need your slides. We're struggling to produce that severe case of enteritis anymore because of all those improvements that we've made over the last 20 years. Eventually, I think this model could really be applied to salmonids as a whole. So trying to get more Atlantic salmon in there as well. And then this slide kind of just gets down to what I've been harping on the whole time here is that enteritis alone is not enough. As we move towards, the advances of diets for the future in aquaculture, we're really looking at subtle changes to intestinal phenotypes between these novel ingredients. No longer are novel feed formulations causing severe perturbations. They're changing very minor biological outcomes. And so I think the model potential is huge here. It's going to reduce that burden, the turnaround time, give us a lot of insights, but we need to develop a model that's a lot more granular and able to segment out those various biological kind of parameters of a histology slide. And with that, I want to leave a little bit of time. I know we took up a good bit of time, and that's completely my fault. But I do want to acknowledge again, Soy Aquaculture Alliance, the U.S. Soy Checkoff Board, been huge to supporting this work. The USDA Agricultural Research Services, the many collaborators at the University of Idaho, and beyond.
Thank you, Jake. I want to first of all, again, thank you for everyone and being here. And there's plenty of opportunity to ask questions. I know we only scheduled an hour here, but let's not force this across the end. I'm curious, Jake. In the last when this project originally started, a lot has evolved in the AI space and open source tools and things like that. Is there anything you would have done differently, knowing all of the different tools now? It sounds like you wrote the majority of the model. Would you tap into, or is there additional advancements you can make by using some of the new sources and models that are out there from, name your favorite AI tool?
Yep. Yeah, absolutely. That's a great question. So certainly things have just continued to advance. I think maybe the biggest thing to consider is there's been a few more of these pre-canned or pre-chained visual models that have come out. So I mentioned earlier that I used a ResNet 18 model. I think it's still a very valid model. In a lot of ways, might argue that it's still probably the best model for us to use. But since the time of kind of proposing this work and diving into it, of course, in academics, once you start research, new tools come out, but you're already halfway through it and you'd be starting all over to go back. But there have been some new visual models, YOLOs and other things. But again, I would argue that the ResNet that we used is probably actually still the most appropriate just because some of those other computer vision models are really focused on different things. So they may be better at recognizing a cat versus a dog
Mhm.
in a picture, but I think the ResNet model still has a lot of advantages on the histology training side. Um, I think there's been some better advances on the human histology side of things. They now have some digital histopathology tools that are quite advanced for human medicine. And I think that's probably the biggest thing that I missed out on taking advantage of with the growth of technology throughout my project is I just didn't have the time to get caught up to what the human world's doing. But I think we could probably borrow a lot of their tools instead of trying to reinvent it for our world. And that's something I've worked a lot with our collaborated histopathologist. Like I said, Dr. Sal Frasca. We've talked a lot about some of those Apereo tools that have come out and working with QPath. These are all the different pipelines that the histologists use to assess these slides. So certainly more advances, and I think some of that would make these model improvements that I suggested at the end kind of quicker to obtain if we were to turn around and tackle this again.
And if I'm playing this back, one call to action, send your slides. So I don't know how we can help cast a broader net for you there, but I'm curious, and this is largely focused on salmonids. What's the next species and application of this technology that could have a huge impact beyond just your neck of the woods in the upper Northwest?
Yeah, the original motivation was to make this a salmonid broad classifier. Unfortunately, the data we were able to get was just rainbow trout, and really, Easton, that's the biggest weakness of this whole project was just not having more original slides, not having more severe cases. But again, I kind of see that as a good thing for the soy side, is just it shows that that broad severe enteritis phenotype is not like what it was 20 years ago. It's very hard for us to find cases of that.
Can you say that
And
one
that's
more time, extra loud and in writing?
Yep. Soy is an awesome ingredient in aquaculture feeds, and it's honestly difficult to find severe perturbations, and
I'll
that is
send you.
sincere. That's not me patting a funder on the back. It's just facts. Like I said, we looked through all of our internally sourced studies trying to find those severe phenotypes of enteritis, and the best I could do was often, a 2 or a 3 out of a 5 ordinal score category. So that's a big thing. I think one of my new insights is that again, we just enteritis, yes or no, is not good enough for the modern world. We need to get a lot more granular with things. In terms of species, I started to say there the original intention was to be salmonid broad, so we wanted to have Atlantic salmon slides in there. I think Atlantic salmon are a clear, front runner to have included here. Like I said earlier, a lot of our more omnivorous species, like tilapia or carp, that are cultured, at high levels throughout the world, they already have no problems with soy. Their more omnivorous physiology of their gut tolerates it quite well. But those carnivores have some of those issues, like Marina was talking about in her presentation, with the anti-nutritional factors, binding out specific nutrient uptake, causing non-specific barrier integrity issues. It's the more carnivorous species that have those issues. So Atlantic salmon be a clear front runner, and then there's growth and continued interest in growing the Pacific salmonids, things like coho and chinook. There's not a lot of classical commercial aquaculture for those species yet, but I hear an uproar of folks that are interested in trying to culture coho and chinook. So again, I think just making this salmonid broad would be the best target for next species to focus on. And again, just need more slides. We've already got the architecture. I can just shove in new slides and update the training on this, and all the computer architecture is already there.
Excellent. And so back to you, Marina. Can you expand on, I know your focus on anti-nutritional factors for this project. Could you maybe rank and share your I know there's less enteritis what's the other causes or what's the next box or focus that we should have in the anti-nutritional space for soy?
Mm. Um, I guess that's kind of a I guess I have to say it's a complicated question. With ANS, we still know very little about them. It's interesting with, I'm going to use saponin as an example. From what I've read so far, saponin tends to be the most potent ANF, resulting in intestinal dysfunction or nutrient absorption, but it also resulted in results that we got that were kind of different from what we read before. And then, of course, with soy being treated and you get soy protein concentrate, soy protein isolate, you have ANFs that are heat labile and heat stable. So that also kind of affects the potency of it, I guess, on how further processed you make soy, which also affects the interactions of the ANFs. It's a difficult question. Yeah, I have to kind of answer it like that, which is what I'm hoping to answer with my program and with our experiments, and if we can get more information on how all that works. Um, I hope that
Yeah,
answered it.
no, it's I know I kind of expected it to be complicated, and obviously your technical expertise is far deeper than mine. So I am excited to continue learning from you.
Well, let me just
Um,
add in there real quick, Easton. So Marina's update here is really kind of, an early prelude to coming results. So she has more work. Her focus that she showed you so far today was on the saponins, but she will look at additional anti-nutritional factors, and I think a lot's going to come out from this new tool, this new perspectives that we're getting from that oosing chamber work that she's doing. So I think she'll have a better answer for you at the end of this project, probably too.
all right, same time next year then.
Yep,
Yep,
same bat channel, same
let's
bat
do
place. You got it.
it.
Perfect. So maybe we'll end on this question of just really what's from your expert perspectives, what's the future for soy and aquafeed? From today, maybe the next 5 years, what's the future and what's our focus need to be?
Yep, I'll jump in just real quick. I think soy has a very sustained place in aquaculture from my perspective. It's really a required, I shouldn't use the word required, but it's a part of all of the formulations that are used commercially nowadays. We have like our program where it's 50% of the diet nearly from the different components of soy that we're using. There are a few commercial producers that are already at very, very high similar levels of soy in their diets. But even those that aren't at a complete plant protein based diet, they're still using a significant amount of soy in their formulations. So I think soy is here to stay for sure. No question about it. Continuing to partner with some of the new species that come on board. One thing we lack in aquaculture is really just the amount of generation time that we've had on our species. As we think about bringing in new fish species, they've not had the same, generations of decades and really even centuries of selection like what we've had in cattle or poultry to get those advantages. So as new species come online, I think there's going to be a lot of work for soy to continue to get them more amenable. I think our programs build a really nice model of how to do that from both the ingredient side as well as the fish side. So just trying to stay relevant with the new species that come on. And then as we continue to work on the research that we're doing at our lab and really throughout the globe, continuing on anti-nutritional factors, looking at different processing methods, trying to figure out ways just to stabilize and improve the inclusion levels. And we've honestly kind of gotten to a point that I think, soy might have reached its apex, or I think there's room for a little more growth, but any formulation, you're not going to want a diet to be 100% anything. So
Yeah.
soy's already, like I said, got a pretty sustained seat at the table for aquaculture diets. It's just continuing to kind of optimize. And that's what I mentioned about my model is no longer are we looking at the big changes. We're trying to make little baby steps of improvements now. So.
Excellent. Marina, anything to add?
I was just going to say positive outlook for sure for soy. Yeah, for sure be able to handle it so well right now. So yeah, just positive from there. Hopefully we can understand more mechanisms on how it all works. Again, coming from a biased point of view, but that just adds on to, how soy affects intestinal function and health and growth and we can answer those questions better. So yeah, definitely positive.
And one final note too, Easton, just this isn't the side of the world that Marina and I are on, but I think on the plant breeding side, there's still a lot of work that can be done too. I mean, like I said, we've seen huge advances already that we've tried to do in parallel, but continuing to work on the soy selection side of things to reduce anti-nutritionals, increase protein. A lot of that work has already made huge gains. We're kind of, like I said, we've made big gains in the last 20 years, so the gains are starting to reach their limiting returns. But again, focusing on just the continued work on improving the soy genetics as well. But we're already at a great place.
Excellent. Well, thank you very much. Pretty quiet group here on the quick Q&A side, so must have been a thorough discussion. But I guess happy Canada Day to you, Marina. Happy 4th of July to the rest of the folks here, and yeah, just really grateful for everyone making time on this afternoon to talk about our research and look forward to our continued collaboration with University of Idaho in this great work. So thanks to both of you for making time and all the prep and to the marketing team behind the scenes. We'll get together again. I believe the next webinar will be in September. So tell a friend and tell a friend to send Jake all of your slides. All right, fair enough. Y'all have a great
Yep.
day.
Thank you.
Appreciate SAA and everyone's time very much. This has been a great collaboration and look forward to continuing research with you, Easton. So thanks for the opportunity to talk to
Yeah.
everyone today.
Thank you very
Yep.
much.
Thank you.
All right, take good care.